Hey everyone, today in this video I'll be talking about dye. Okay, now dye, that is very much important in case of microbiology. Why? Because these type of dyes that are used to stain the bacterial cell, okay, and we can see them under microscope, under light microscope. So dye, what is the dye? The definition of dye is basically it's an organic compound which can color the bacterial cell. Okay, now the dye that has two important but different groups, one that is known as chromophore group, okay, this is one, and number two, the second group, which is also important, is known as auxochrome group. Okay, so one that is chromophore, one that is auxochrome group. Now what is the function of chromophore group? Chromophore that actually imparts the color property of dye. Okay, any kind of a dye like methylene blue or gramsidine in case of crystal violet or saffronin, the chromophore is basically important to impart the color property of a dye. Okay, and what is the function of this auxochrome? Now auxochrome is not directly involved in the color intensity of a dye. Basically auxochrome that will helps to bind the dye to the material or the bacteria. In case of microbiology, to the bacteria, the auxochrome that will help to bind the dye particle to the microbes. So these two are very important. One thing that is very much important, you should remember that if a dye that doesn't contain any kind of a auxochrome, only chromophore, then it only has the colors property and known as chromogenic substance or chromogen. But without the auxochrome, any kind of organic compound which has the chromophore group, they cannot bind to the bacteria or any kind of a material. So auxochrome is very much needed for that. And the first organic dye that is known as aniline, okay, first invented as organic dye in case of microbiology. So this is it. Thank you so much.